A very good morning, good evening to one and all present today. Welcome to the first episode of the Infectious Disease webinar series hosted by the Euroimmun Academy. Today, we would be discussing a very familiar topic, a well known topic to all of us, that is the global state of COVID 19 diagnostics and the challenges that lie ahead of us. Some housekeeping rules before we begin. Similar to our previous webinars, we have two speakers presenting today. Towards the end, we will also have a Q&A panel discussion. If you have any questions, please type them in the chat box that you find on the right side of your screen. And we would be moderating your chat so that we can relay it to the speakers. Any unanswered questions will be separately addressed via an email. Without further ado, it is my pleasure to introduce you to our first speaker, Dr. Brandon Michael Henry. Dr. Henry is a physician scientist at the Cincinnati Children's Hospital Medical Center and a world renowned expert on COVID 19. To date, he has published over 100 articles in international medical journals on SARS CoV 2 infection, and these have already been cited over 6,000 times. Dr. Henry is the leader of multiple international studies investigating COVID 19 infection, and he has also helped establish the international guidelines for COVID 19. His research has also focused extensively on unraveling COVID 19 pathophysiology, elucidating the mechanisms driving COVID 19 associated coagulopathy, immunopathology, and multi organ injury. Additionally, he serves as an international advisor on COVID 19 response to multiple regional and national public health agencies. We welcome you, Dr. Henry. I will now pass it over to you to begin your presentation whenever you're ready.
Thank you for the introduction. I'm excited to be here today to talk to you about the global state of COVID-19, the diagnostics and the challenges ahead. Over the next 45 minutes, we're going to dive into a whole bunch of important topics related to the pandemic, many of which are rather controversial. But before I do, I just want to give a shout out to my affiliation, Cincinnati Children's, the Clinical Nephrology Lab, led by Dr. Stephanie Benoit, our lab manager, Thelma Kathman, our amazing research staff, especially James Rose. They've been the backbone of my research and really been at the forefront of serology testing throughout the pandemic. And to Texas Biomedical Research Institute, which I recently joined. They've done some of the most incredible work in the world on COVID-19. We also house here the Southwest National Primate Research Center, which was where they did the preclinical trials on the mRNA vaccines to Pfizer and Moderna. And if it wasn't for them, we'd never been able to get the clinical trials and get the shots into your arm so quickly. So I'm very fortunate to be part of both of these great places. So here's an outline of today's talk. Each one of these topics that we're going to go through really could be a whole separate talk in and of themselves. So if there's any topic of which I don't go into enough detail, feel free to ask a question or send me an email later on, and I'd be happy to discuss it with you. We're going to talk a little bit. I'm going to start with talking about the molecular basis for laboratory testing, what you need to actually know about the virus to understand it. What about variants? We're going to talk a little bit about variants. We're going to go into an overview of the different types of diagnostics we're currently using and the future directions. We're going to talk about vaccinology and assessment of vaccine efficacy with serology, T-cell assays, and evaluating B memory cells. We're going to talk about, then we're going to dive in, I should say, more into serology testing, because that's really the bread and butter. We're going to talk about why, when, and how. We'll talk a little bit about the advantages and the drawbacks of serologic monitoring of vaccine efficacy. We're going to discuss throughout the lecture boosters and breakthrough infections, and then go into what lies ahead and how we can be prepared for the future. I have no disclosures, no conflicts of interest, but I always put a warning label whenever I talk about COVID-19 that anything I say today can be wrong tomorrow. It's just the nature of the fast-moving science that we have during this pandemic. So let's talk a little bit about the virus, give a little bit of introduction. SARS-CoV-2 is an enveloped single-stranded RNA virus. It has four main structural proteins. the membrane protein, the envelope protein, the nucleocapsid protein, and the one we hear most about, the spike protein. There are other important proteins, we call them the non-structural proteins encoded by the open reading frame region that is not shown. These include things like the viral polymerase. They don't get as much attention as, for example, the spike protein, but it's very likely that mutations in some of these non-structural proteins likely play a role in the pathology of different variants. So here we have the structure. We're going to start with talking a little bit about the spike protein because it's an antigenic target for many assays. The spike protein is what enables host cell attachment and fusion. The S1 subunit of the protein, which you can see here, contains the receptor binding domain, which binds to the host angiotensin converting enzyme receptor. Neutralizing antibodies block the binding of the virus to the ACE2 receptor. Now, it's important to note that neutralizing, the definition here that we're going to use is any antibody that blocks the binding. So as we'll talk about later, it doesn't necessarily have to be an antibody that blocks the receptor binding domain. It could just do any function that stops the virus from being able to bind to ACE2. And that's a very important definition that we'll come back to. The nucleocapsid protein is responsible for the replication cycle. It's responsible for packaging the viral genome, viral assembly, and also plays a role in modifying the host response to the virus. The membrane protein promotes stabilization of the viral envelope and interacts with the envelope protein to release viral particles, demonstrating that it's quite important in complete virus production and maturation. Going back to the angiotensin converting enzyme 2 receptor, ACE2 is an important enzyme in the renin-angiotensin-aldosterone system pathway. It sits on the surface membrane of epithelial cells. The RAS system that we call it, the renin-angiotensin-aldosterone system, plays a key role in the regulation of fluid balance, electrolytes, and many other pathways, especially blood pressure. And it's long been thought by me and many others that the way the virus interacts with the ACE2 receptor indeed leads to a significant proportion of its pathophysiology. The ACE2 enzyme receptor is widely expressed throughout the body. We can see here an expression profile. You can see that it's quite widely expressed in the lungs, but in a lot of tissues like the testes. You can start to think that, we hear a lot about males having worse disease. Well, some of the highest expression of ACE2 is in the testes, but also the intestines, adipose tissue, kidney, heart, thyroid. So it's really a widely expressed enzyme. So here we could see an article that's published in the European Respiratory Journal. And I just wanted to point out here that ACE2 expression is highly variable and it's impacted by so many different factors. For example, smoking and COPD can increase the levels of ACE2. And I think that we'll see in time as more research is done that ACE2, the differences in ACE2 expression between individuals plays a large part of why we see, some people get really sick and other people, have mild or asymptomatic infections. It's probably not the only factor, but the genetics behind this likely plays a role. So let's talk now about variants. Now that we have the virus defined, we know the parts of the virus. What's a variant? So we're going to use some definitions here from the CDC. A mutation refers to a single change in a virus's genome. It happens very frequently, only sometimes changes the characteristics of the virus. Most of the mutations that we see are harmless. They're, we'll call them silent, they're synonymous mutations, and they do nothing. It just codes for the same, it's upcoding for the same amino acid. On the other hand, it could create a virus, that viral particle that's just not functional. So most of the time when a mutation happens, it very rarely changes an important characteristic of the virus. A lineage is a group of closely related viruses with a common ancestor. SARS-CoV-2 has many lineages. They all cause COVID-19. And now we're hearing a little bit, for example, in the UK about a new variant associated with the Delta variant. So that's from that particular lineage. A variant now is a viral genome that may contain one or more mutations. In some cases, they're a group of mutations with similar genetic changes, such as a lineage or group of lineages, may be assigned that we'll see, you've been hearing a lot by public health organizations that we call variants of concern or variants of interest due to shared attributes and characteristics that require special public health action. The WHO defines a variant of concern as an increase in any variant that causes an increase in transmissibility or detrimental change in COVID-19 epidemiology, an increase in virulence or a change in clinical disease presentation, or any decrease in effectiveness of public health and social measures or available diagnostics, vaccines, and therapeutics. And the CDC currently has 4 that are labeled variants of concern: the Alpha, which was the UK variant, the Beta, the South African variant, Gamma, which arose in Brazil, and the Delta variant, which is now predominating throughout most of the world, which arose in India. The CDC now has modified their definitions, so they now have 4 categories to define variants. We have variants being monitored, and that includes right now in the United States, Alpha, Beta, and Gamma. Then they have variants of interest, which require more special study. There are currently none of those being monitored by the CDC or that fit that definition. Then we have variants of concern. These are ones that are being closely monitored and characterized by federal health agencies. And this includes right now the Delta variant. And variants of high consequence, which cause significant impairment to vaccine efficacy or an increase in disease severity. And there are currently no SARS-CoV variants that rise to that level of high consequence at the moment. So here we can see, for example, how the CDC will describe the Delta variant. We have over here the description of the spike protein substitutions, and as you can see, there are several. They provide where it's first identified, and the reason that it's been given that characteristic is because it's been associated with significant increased transmissibility. We'll talk about that in a second. A potential reduction in neutralization by some monoclonal antibody treatments, and potential reduction in neutralization by post-vaccination sera. So what's the biggest problem we have with variants? Well, the biggest issue that we've had with variants so far is that they seem to be all associated with increased transmissibility. And that causes significant problems when we're talking about trying to achieve herd immunity and getting back to normal life. We describe the transmissibility of a virus using something called the basic reproduction number, right? And the basic reproduction number is the number of people on average infected by a single person with the disease. So for example, with influenza, on average, a person infected with influenza will infect 1 to 2 other people. When we look at COVID, we can see that the original, which we call the wild type, right, variant that arose out of Wuhan, had a basic reproduction number of around 3. And we can see that the UK and South African variants, they were up to a number of 5. Here's polio. And then for the Delta variant, it's not even on the chart. It's over here almost near 8 or 9. And as you can see, as we go up in the transmissibility, the more infectious the virus is, the higher threshold of herd immunity we have to get. So here we are, I think, in sort of a currently challenging position. We have variants that are increasing in transmissibility, which increases the threshold for herd immunity. And as we get away from those initial vaccine doses, immunogenicity or vaccine protection sort of drops. And we get to a harder and harder point trying to achieve herd immunity. So let's talk about COVID-19 testing. We have 4 main types of tests that we'll talk about: molecular, antigen, serological, and the IGRA, which we'll call the interferon gamma releasing assays. Molecular tests are sort of the gold standard. The diagnostic window is during an active infection, though, as you probably have heard, many cases that you can sometimes detect RNA or the viral shedding for many weeks after infection. Its aim is to detect the pathogen. It's highly sensitive and specific, but it cannot distinguish between infectious and non-infectious virus. For that, you would have to do like a plaque assay. Just because it's the gold standard doesn't mean that there's not issues with it. Just this past week, there was a report in the BMJ News that a major private laboratory in the UK had to stop their PCR testing, molecular testing, due to a high rate of false negatives. So quality control is always important. We have the antigen test. These are commonly referred to as the rapid test. The diagnostic window is active infection. I was just going to highlight that usually these perform best in patients who are symptomatic. They have a slightly lower sensitivity than molecular tests, and false positive results can occur. Most of these are designed to detect the nucleocapsid protein, which is important. We'll talk a little bit about how variants impact testing in a few minutes, so keep that part in mind. Next, we have serological tests. These detect convalescent infection. They aim to detect antibodies. We can use them to monitor immune response, provide information on vaccine efficacy. However, the role of antibody duration and the titer are not fully understood, and we'll talk a little bit more about that later on. Last, we have the interferon gamma releasing assays. These are designed to assess T-cell response. Robust responses by T-cells have been reported to both viral infection and the vaccine. And patients with undetectable antibodies often do have a detectable T-cell response. However, the role of T-cells and the clinical significance of these tests require further investigation. So here we can see sort of an outline of the serological response. during the course of infection. We also have here RNA. We see that RNA is the first thing that we could detect, followed by antigen. RNA tends to persist a little bit longer, it can persist for several weeks in some cases. In terms of antibody responses, we first will see that it's IgM and IgA. IgM, I think, as many people now know, has a very variable pattern. And it's not always detected in some individuals. It tends to decay very fast, as you can see here, even at 14 days after symptom onset, it's really starting to decline. IgA tends to persist for like 2 to 3 to 4 months, depending on the study. T cells also begin rather early at that same time point, about 3 to 5 days. Around 10 days on average, I think you can start to see 7 to 10 days, the IgG response, and the IgG response has been known to persist for several months now. And we'll talk a little bit about that later. So what's the impact of variants on COVID-19 testing accuracy? I think this is one of the most important slides of my presentation. Antigen test, there's probably a very small marginal risk that variants would impact that. I'm not saying it's impossible, but I'm saying currently it's marginal. And the reason for that is they're designed mostly to detect a nucleocapsid protein, and the nucleocapsid protein tends to be very conserved. There's not that many mutations associated with the nucleocapsid protein that would change its structure and leave it undetectable by an antigen test. Molecular test, a little bit higher risk, so I'd say small risk. Now, most molecular tests on the market, they usually PCR for at least 3 genes. That's not to say that they can't be affected. As a matter of fact, during the outbreak with the UK variant, the country was able to actually trace the variant, because of the fact that the spike protein was dropping out. It wasn't being detected on PCR, but you would have the 2 other genes, because you're testing for 3, coming up positive. So that particular pattern with the spike dropout, but 2 others positive, and we usually use the structural proteins, sort of was an identifier for that particular variant. Serology is a little bit more complicated, right? And I put small to moderate risk. And I think it really comes down to, how are we using serology? Are we using it to detect past exposure to an infection? With a variant, in which case, there's probably a smaller risk, or are we trying to use serology really to predict neutralizing capacity to protect against the variants, for example, in someone who's vaccinated? And I think that gets a little bit more complicated. That's a little bit more tricky. And we'll talk a little bit more about that. In terms of the T cell assays, the interferon gamma releasing assays, we're not really sure. I haven't seen enough data yet to really draw any type of conclusion or risk assessment in terms of how that impacts variants, but I would imagine that it probably is a little bit similar to serology. The FDA does maintain a website where they report about the impact of viral mutations on COVID-19 testing, and I do recommend you check it out. They also have a specific policy and guidelines for evaluating the impact of viral mutations on COVID-19 tests. There's been a few studies now looking at the impact of variants on commercial serology assays. And for the most part, this one preprint has not been peer reviewed, but it did conclude that even though there was some deterioration in correlation between serological measurements and functional neutralization activity, some assays maintain the ability to predict neutralizing titers even against variants of concern. So I think when it comes to, saying, predicting neutralizing capacity, it's a little bit trickier. Where I do think that when it comes to just detecting a prior infection, these assays probably perform very good, because you don't need to, the titer is less important when it just comes to, say, identifying past exposure. But when it comes to, saying, predicting that exposure from the titer, it's a little bit more complicated. So let's now talk a little bit about how testing can play a role in optimizing the effectiveness of COVID-19 vaccination. But before we do that, let's define, how do the vaccines work? In general, all vaccines work by utilizing the natural defenses of an organism for developing a sustained resistance to specific infections, ultimately strengthening the immune system, which responds to vaccination in 1 of 3 ways. 1, producing specific antibodies, and that's called the humoral immunity. 2, training immune cells to combat the pathogen and infected cells that generate mediators for amplifying the immunological response, and we'll call that cellular immunity. And 3, triggering the generation of memory cells, which will especially help to face recurrent infections. So the mRNA vaccines, these are the ones most commonly used around the world. We have Moderna, Pfizer are the big names here. And they basically work by injecting mRNA right into your arm. The mRNA gets uptaken by the cells. The cells then translate that mRNA into spike protein, right? So we're only producing the spike protein with the mRNA vaccine. It's important to remember. The spike protein is then recognized by the immune system as a foreign invader, and we then develop antibodies against that particular spike protein, so that if you're exposed to the virus, you already have antibodies that bind to the spike protein on the virus, as you can see here, and hopefully neutralize it. So going back to that adaptive immunity, because we're going to talk a lot about measuring this, we have two main components of adaptive immunity. So we have the humoral arm and the cell-mediated arm. The humoral arm is mostly, we're going to talk about the B cells here. B cells are the cells that really produce antibodies, right? They become plasma cells, and these plasma cells produce large amounts of antibodies, most of which we hope are neutralizing. And then we have T cells. We have two types of T cells. We have the CD8 T cells. These are also called the cytotoxic T cells, and they're designed to kill infected cells. And then we have T helper cells, and these play a role both with cell-mediated and humoral immunity. And there's many types. They can activate the immune system. They can suppress the immune system. Here we can see on the right side of the panel, we have an antigen-presenting cell. Right, these are the cells that will go to these lymphocytes and say, hey, I found this spike protein, or I found this part of the virus, do something about it. All right, and the cytotoxic T cells will then go around, try to identify infected cells, and they'll attack and kill those infected cells. And it'll also tell B cells, hey, start releasing that neutralizing antibody, right, so that the virus can't bind and infect any more cells. In general, vaccine efficacy can be assessed by two means. So clinical efficacy and biological efficacy. And both really do involve some component of laboratory testing. Biological efficacy can be considered a surrogate approach for predicting and verifying vaccine efficacy, aka immunogenicity. Biological pathways work in synergy to protect a subject from being infected or reinfected, as well as to mitigate virus-induced local and systemic injuries, thus leading to a decreased risk of morbidity and disability and death when infected. Although laboratory measures are only surrogate endpoints of vaccine effectiveness, their assessment provides a basic advantage. It does not require these long prospective monitoring periods like we did with the vaccine studies, enable monitoring of real-world vaccine immunogenicity, which may provide real-time information to better tailor interventions for both single individuals and the population as a whole. So we can see sort of a diagram of our two routes for assessing vaccine efficacy. When it comes to clinical effectiveness, we can monitor for newer breakthrough infections, which is something, unfortunately, the CDC decided in April not to do. And I think that was a mistake. We should have done it. We should have measuring titers in these patients so we understand what threshold of antibody is, the point at which we start to see breakthrough infections in different populations. We can measure viral load in infected patients. We can monitor for hospitalizations, use of mechanical ventilation or ICU admission in vaccinated individuals, or deaths. The con is that these tend to be longer perspective, and they're certainly very much impacted by time. I think if you go back a bit, when we're talking about some of the vaccine trials, Pfizer and Moderna showed really high efficacy early on, but that was when the earlier strains were circulating, where Johnson & Johnson came out a little later and showed not as high levels of protection, so the clinical efficacy wasn't as high, but at the same time, they were facing a whole, slew of new variants that were out there at the time, including the UK and South African variants. On the other hand, we can use biological measures of efficacy, right? And these include assessments of humoral immunity with neutralizing antibodies and serology assays, assessment of cellular immunity, which we can measure CD4 and CD8 T cell responses using like the interferon gamma release assays, and assessment of immunological memory. So memory Bs or T cells, like we said, the B cells can be plasma cells. They can also become memory cells. So upon re-exposure to an antigen, so if you get reinfected later, we're able to produce antibodies more quickly. And this can be assessed via flow cytometry. So on the other hand, where clinical effectiveness takes a long time, biological effectiveness can be real time. It can be prospective. It can be cross-sectional. So it gives us a much faster assessment of immunity within the population. So let's talk about these three forms of measures of biologic efficacy. SARS-CoV-2 serology is now universally considered a key aspect in assessing vaccine immunogenicity. Although a kaleidoscope of anti-SARS-CoV-2 immunoassays have been commercialized, evidence now suggests that measuring serum levels of total or IgG class antibodies targeting the SARS-CoV-2 spike protein or its receptor binding domain are those that best correlate with endogenous viral neutralization potential. Recent evidence has been published that vaccine breakthrough, defined as detection of SARS-CoV-2 RNA or antigen greater than 14 days after the receipt of a recommended dose of authorized COVID-19 vaccines, likely depends on the serum concentration of anti-SARS-CoV-2 antibodies. And so here we can see a study showing this, right? That so they measured the antibody levels, vaccinated healthcare workers, and those who were infected, who developed infections, these breakthrough infections. And they basically found that the level of anti-SARS-CoV neutralizing antibody and anti-SARS-CoV IgG were over 50% lower in breakthrough infected subjects than in unmatched infected controls. Overall, the peri-infection value of SARS-CoV-2 neutralizing antibodies was associated with a lower viral load, as reflected by a higher cycle threshold values on PCR. So the higher the level of anti-SARS-CoV-2 antibodies that a patient had when they had a breakthrough infection, the lower the viral load was overall. Similar evidence has been reported in another investigation where it was found that vaccine efficacy against primary symptomatic COVID-19 was directly related to the anti-SARS-CoV-2 level of anti-spike IgG, anti-RBD IgG, and neutralizing antibodies achieved after receiving an adenovirus-based vaccine such as Johnson & Johnson. These findings converge really to suggest that vaccine breakthrough, at least in terms of failure to prevent the development of an active viral infection or reinfection, may be strongly dependent on the level of anti-SARS-CoV-2 antibodies, such that repeated monitoring in subjects whom immunogenicity would be expected to be lower, and we'll talk about those subjects, might be highly advisable for establishing a program of vaccine stewardship. We're going to come back to serology testing in a bit more detail, but let's jump a little bit to assessment of cell-mediated immunity. So we did humoral, serologic. Let's go to cell-mediated and T cells. T cells generally play a crucial role in the adaptive immunity for viral clearance. SARS-CoV-2 T cell responses can be detected about 3 to 5 days post-symptom onset, and functional SARS-CoV-2 specific T cell responses have been seen up to 6 months following infection. T cell responses against the spike protein do correlate well with IgG and IgA antibody titers. High levels, though, of some cytokines seen in COVID-19 patients as part of the systemic inflammatory response, the hypercytokinemia, may prevent the development of long-term immunity to SARS-CoV-2. This phenomenon, which we'll call immune amnesia, is unlikely to occur after vaccination, really suggesting that vaccination is definitely the best way to generate effective herd immunity. So when you hear people say, well, I've been infected, I had COVID-19, so I don't need to get the vaccine. Well, no, because you may not have had such great T cell responses, and T cells play an important role, likely play an important role, I should say, in the overall immune protection that you get following vaccination or an infection with an infectious disease. It's been reported that about 20% of patients who recovered from SARS-CoV-2 infection without detectable antibodies showed a T cell response against the virus, which suggests it may be important to measure both B and T cell mediated responses post-natural infection, as well as in vaccinated individuals. More research, though, is really needed to assess the role of T cells in COVID-19, as well as how strong the T cell response has to be to provide protection and for how long that protection lasts. Therefore, I think it's safe to say that the current use of assays assessing T cell response should be concentrated in research use, not in routine clinical testing as of now. That said, interferon gamma releasing assays have already been used reliably for monitoring cellular response in COVID-19 patients and for tailoring treatment. The assessment of T cells can be performed using a variety of assays, for example, ELISpot. We talked about interferon gamma releasing assays, and there's a variety of other tests using flow cytometry, next generation sequencing, etc. A couple of things with T cells. There was one interesting study. This was done by the NIH, where they took macaque monkeys, and they wiped out their T cells, so they were T cell depleted mice. It was interesting, though, that wiping out the T cells did not induce severe disease, right? So the disease was the same in the T cell depleted and T cell efficient mice, efficient macaques. And it sort of raised the question about, well, what is the role of T cells in COVID-19, seeing that wiping out T cells didn't result in more severe disease. We talked a little about that immune amnesia. So when you have an infection, that your immune system becomes a little bit suppressed and you don't develop that long-term immunity. When we talked about the cytokine storm, we looked at this particularly, and we found that the interleukin 10 is a strong predictor of severe disease and poor outcomes. We did a study at Cincinnati Children's, and we found that both looking at IL-10 and IL-10 lymphocyte count ratios was more associated with predicting severe outcomes than was looking at the pro-inflammatory response, such as interleukin-6 and interleukin-8. So certainly immune suppression plays a big role in COVID-19, probably doesn't get enough attention. I actually talked about this really early on. We see a lot of patients who develop lymphopenia and COVID-19, and recovery from lymphopenia probably is a very important factor in long-term outcome, as well as the ability to generate effective immunity. Studies looking at patients who recovered from COVID-19 has also shown that patients, a good number, I think in this study, they found 14% of patients who recovered from COVID-19 had lymphopenia at a follow-up range of 80 to 100 days. So this type of immunosuppression can last for quite a bit of time and likely then impairs the ability to develop an effective immune response. Now we're going to talk about memory B cells. That's the third monitoring. Recent data has shown that the kinetics of memory B cells producing anti-SARS-CoV-2 neutralizing antibodies made it their special focus. Although sustained generation of these types of cells may not be fully effective at preventing vaccine breakthrough, it may be instead effective at reducing the likelihood of developing symptomatic or even severe disease due to an earlier antibody response in patients who have B memory cells. And this is probably most important, really, when the level of anti-SARS-CoV-2 circulating antibodies has declined. Right? So this one study showed that even in patients, serum from patients that have been low antibodies at the 7 months after vaccination, there was still value in memory anti-spike protein, anti-receptor domain B cells that remained relatively high, thus suggesting that the immune system then is somehow primed to face new SARS-CoV-2 infections in vaccinated individuals. So that doesn't mean. So I think the real point of this is that though the antibody titers have declined, hopefully that in those patients, well, they still might get infected, they might still have symptoms. The B memory cells, the earlier immune response because of the B memory cells, should at least help prevent against severe development of severe COVID-19, needing, for example, mechanical ventilation, ICU admission. And that includes some protection against the variants of concern. So now let's move on to focusing going back to serology testing. Again, that is really the bread and butter of vaccine efficacy monitoring. And we'll talk a little bit about the why, when, and how of vaccine efficacy monitoring using serology. So what is the why? Why do we need serologic monitoring programs? If it was me, we would have nationwide serological monitoring programs. I think that there's two main reasons: patient factors and variants of concern. Certainly there are others, but I'm focused on these two. And the first is inter-individual, the immune response has a lot of variability between individuals. Recent population studies conducted on baseline seronegative subjects, so those who have never been exposed, never had COVID-19, revealed that immunogenicity after mRNA vaccination differed widely between recipients, with variations of neutralizing antibody levels extending between 1 and 2 orders of magnitude. And these studies were done by myself and Giuseppe Lippi in Verona, Italy. And we found that the level of the antibody response after vaccination is impacted by a large number of factors, including age, sex, and BMI. And BMI is really interesting. One study by Pellini and colleagues found that after Pfizer, levels of anti-SARS-CoV-2 spike IgG antibodies were between 50 to 60% lower in pre-obese and obese individuals. And moreover, vaccine-elicited immunogenicity has also been found to be considerably attenuated in many specific populations of patients with comorbidities. And the number of comorbidities, as you see, is quite wide and probably covers a large part of the population. And then variants of concern, right? We're not quite sure what level of antibody titer is efficient for protecting those variants of concern, but we do know variants of concern are associated with a decay in the neutralizing potential of the antibodies generated. Right? So, for example, with the UK variant, we see a 1.7-fold reduction, with the South African variant 7.9-fold, with the Brazil variant 5-fold, with the Delta variant 6.8-fold. So many of these are associated with significant reduction in neutralizing potential, which ideally means we should be monitoring patients to, make sure that they're maintaining an adequate titer. This also means that some of the new monoclonal antibody treatments that are available likely have suffered from some decline in efficacy due to decreased neutralizing potential against the variants. We still don't know much about how variants of concern impact immunity, right? What is the threshold of decay in neutralizing potential that is associated with breakthrough infections? We don't know because we don't have a nationwide monitoring program. What's the role of T cells? Again, we're not quite sure, and that requires further studies. We also need further studies to really clarify whether mutations outside of the receptor binding domain. So we talked about how spike has the different parts, and we have S1, S2, and on S1 we have the receptor binding domain. How mutations outside of that receptor binding domain in the spike may have a synergistic effect towards reducing the neutralization potential of anti-SARS-CoV-2 antibodies. And finally, what role do boosters play? How do boosters help in protecting us against variants? So going ahead, we talked a little bit about patient-level factors. We did a study, a systematic review, actually looking at this, pooling some of our work as well as work done around the world. This was done by myself and led by Kin Israel and Notarte from the Philippines. Kin is a really budding clinical virologist, and I expect that we'll see really big things for him in the future. So we overall looked at 32 studies that assessed the efficacy of the Pfizer vaccine and measured antibody levels in patients with different conditions and different factors and comorbidities. Due to the significant heterogeneity in the many assays that are out there, we tried to standardize results between different assays by calculating a % difference in means between groups. So this enabled us to create a comparison of laboratory results regardless of the units of measurements and the diagnostic tools for quantification of antibody titers. So we found that there are several factors that were significantly affecting vaccine humoral immune response. We found that, of course, across the board, antibody response of younger individuals is significantly greater than older individuals. And that was across all age groups, regardless of the age stratification. Of course, the difference becomes more pronounced when we have larger age gaps. But I think the real point is that those over 65 have significantly decreased vaccine-specific antibody titers. They're more likely to be non or low responders and more likely to have rapidly waning antibody levels. And the difference in antibody response is more prominent in the first dose. We found this as well as many other groups, but the differences decrease over time, especially after the second dose, but do remain significant. You can see one study, and I think I'm just going to point out this one here, is it's really interesting that in those less than 30, right, as adults, less than 30, so it's a rather, you're looking at like 18 to 30 here. And those, 30 to 39, those less than 30 had 61% higher level of antibodies. So that just gives you how significant these are. And it's certainly variable between different age groups. So less than 30 is 50% higher than over 60. Here, this is our study that we did in Verona, Giuseppe Lippi's group, and we found that, after the first dose, the IgG titer is 84% higher in those less than 60 as compared to over 60. And this decreased to a significant, still significant 33% after the second dose. Another study, again, showing how high, consistently high, so this study was run by Muller, was 87% higher in the younger group after the first dose. I think in the next talk, they're going to go into some of these studies in a little bit more detail. Sex is also a really important factor in vaccine efficacy, with female IgG production significantly outperforming males after the first and second dose. It gets into really interesting physiology here and genetics. The X chromosome in the female provides greater inflammatory, antiviral, and humoral immune responses compared to males, and certainly is a factor why we probably see more severe disease in males. Part of the reason, for example, is receptors that recognize the virus and generate interferon-mediated responses, such as the toll-like receptors. They're expressed on the X chromosome. And in females, they tend to be able to escape X chromosome inactivation. So females can end up having two of these genes actually expressed, where males are only ever able to have one. Estrogen also directly modifies immune response and inhibits SARS-CoV-2 replication by affecting cell integrity, often reducing the incidence of SARS-CoV-2 severity, I should say, infection in females. And testosterone overall has an immunosuppressive effect. And we talked about this in a little more detail. I put these studies on here, these reports that we wrote. So if you're interested in this topic, because it is really interesting, ACE2 expression also probably is different between males and females and plays a role. And ACE2 is also an interferon gamma regulated gene. So it's really a really interesting topic, and definitely you should consider doing a little further reading on this. As you can see here, we assessed this. So in our study, we found at after the second dose, females had about 13% higher titer than males, and these numbers were rather consistent across the board. Serostatus is an interesting one. This really, when we say serostatus, we're meaning whether or not patients were previously infected with the virus or not before getting their first dose. And we found that there is significant individual heterogeneity in these patients. They tend to develop earlier antibody responses and have preservation of their B-cell mediated memory, likely contributing to these responses. But overall, seropositive, so people who were previously infected, tend to have more robust accelerated humoral immune responses after the first vaccine dose. There is some variability amongst this in the literature. It's a complicated topic, and one of the things we actually saw is at 6 months, we don't have the data here, but at 6 months, we actually saw that patients who were seropositive before had a more rapid decline in their antibody titers. So here you can see, just as an example, after the second dose of vaccine, the IgG titer of patients who were previously exposed or infected with the virus is 91% higher than in those who were never infected before. So those tables, by the way, are all they're all online at our preprint on medRxiv, so you can go there if you want to go read into the details. Comorbidities, and there's a lot of these, and we're just going to kind of point out a little bit of the data here. Hemodialysis, right? Patients who receive hemodialysis has a significantly lower antibody response than those who are not compared to healthy controls. That is also impacted by age, and I think all comorbidities are complicated by advanced age. Transplant recipients, they tend to develop a later antibody response that is significantly likely below the protective threshold, and they unfortunately have faster decay in their antibodies. Cancer and immunosuppression, another whole group of patients who have significantly lower antibody responses and faster waning of immunity. And then this one as well, like metabolic derangement, especially obesity. Those who have lower BMI tend to have significantly greater production of IgG antibodies compared to pre-obese and obese individuals, right? And similar to hypertension, right? Normal tensive individuals have better immune responses than those who are hypertensive. I point out a couple of these numbers just so we can see. For example, here, one study by John et al. And colleagues, they looked at healthcare workers versus 60 to 69-year-olds on hemodialysis and found that, for example, antibody titer is 50% higher in healthcare workers. We can see even within hemodialysis patients, it's impacted by age. For those, for example, 60 to 69-year-olds on hemodialysis have 43% higher levels of antibodies compared to those who are 80 to 90-year-olds. So that's definitely a vulnerable group of patients and should be prioritized for boosters. Another study here looking at cancer. These are patients with lymphoma, and they found that IgG titer was in controls is 80% higher than in those with cancer. This study looking from Israel, looking at patients with solid tumors and undergoing active cancer treatment, found that the IgG titer of controls is 68% higher than in cancer patients. And lastly, we'll point out one other is looking at, for example, autoimmune inflammatory rheumatic diseases. They often overall, and there's a breakdown by type of inflammatory diseases in the table, but found that the antibody titer is 39% higher in controls than participants with autoimmune inflammatory rheumatic diseases. In transplant patients, we should point that out. The IgG titer, for example, compared to controls, is 92% lower in those who receive renal transplant. So another group that really should be prioritized for boosters and should undergo serologic monitoring. So here we have an updated list of the major correlates of vaccine immunogenicity. So those patients who have repressed, and you can see it's wide ranging: older age, sex, obesity, cancer, impaired renal function, cardiovascular diseases like hypertension and heart disease, which are extremely common in the population. Any type of immunosuppression, so transplant patients, patients with autoimmune diseases, and drugs. There's a whole list of drugs that are associated. But also infections of variants of concern, right? There's in areas where we have widespread variants of concern being circulated in the population, those patients probably should undergo serologic monitoring, even healthy individuals. So let's move on to the when. When should we consider serologic testing? Ideally, we would have baseline before vaccination levels, especially in vulnerable populations, but that window is a bit past, I think, now. A 1 month measurement would enable timely identification of lower non-responders to vaccination, while the 6 month assessment would allow for recognizing vaccine recipients displaying a faster antibody decay rate. In both patients, then consideration could then be made for administering a vaccine booster. After that, I think we're looking at every 2 to 3 months, but again, it's depending on the specific patient population and what's circulating currently. So we just measured, we finished our analysis, and this was published in the Journal of Medical Biochemistry. We measured total anti-SARS-CoV-2 antibodies 6 months after Pfizer and healthcare workers. And we did find, it looks a little bit deceiving, it doesn't look very like a significant decline, but do keep in mind that these are, tenfold reductions here along the y-axis. We saw that, from the peak after the second dose, we've had significant declines, and I think we'll see a little bit better here what these significant declines look like. Here we have it broken down by sex and age. We can see that women overall have better responses regardless of age, but we can really see that as time goes on, the levels of antibodies really do start to decay. And how much do they decay? So here we have the red is at 6 months. So this is assessing how much they declined from peak. We can see that women less than 65, there was a 56% decline. Women greater than 65, 52%. Men less than 65, 52%. And men greater than 65, 56% decline. So these are quite significant levels. Of course, we don't yet know the threshold for what titer is associated with protection. That's also probably impacted by variants, a very complicated question, but it's significantly concerning. And we're not the only group that found this. This study from Israel also found that there's significant waning in humoral immune responses over 6 months after vaccination with the Pfizer vaccine. And one other study really found that the T cell responses tend to follow the IgG responses, and there was significant decline in T cell responses at 6 months as well. Now comes to the how. How should we do seromonitoring programs? How should we assess vaccine efficacy? And we talked a little bit about it before, but now we're going to really focus on how we should do serology with Serology. There is now solid evidence that the neutralization activity of post-infection or post-vaccination serum mostly resides in antibodies targeting the spike protein or its receptor binding domain. But it's important to point out that anti-receptor binding domains assays should never be considered a measure of neutralizing level. And we talked about that before, and I'll highlight it again, because said neutralizing antibodies are defined as an antibody that binds anywhere on the virus and blocks its binding to the host receptor ACE2. So it's better to employ a surrogate viral neutralization test for assessing neutralizing activity. And the reason, again, I point this out is the study by Voss and colleagues showed that 84% of all neutralizing antibodies target sequences outside of the receptor binding domain, and that those binding the N-terminal domain, so the end of the S1 subunit of the SARS-CoV-2 spike protein, were highly protective in a model of lethal viral challenge. We also found, that neutralizing potential, so neutralizing, we'll talk about neutralizing activity assays, like so the surrogate viral neutralization test, they tend to correlate very well with immune protection from symptomatic SARS-CoV-2. Here we can see, for example, how this type of assay works. It's the Euroimmun assay. So the surrogate viral neutralization test basically works by using a synthetic ACE2 and measuring your ability of your antibodies to stop ACE2 from binding to the receptor binding domain on S1. So if we're only, though, using a single immunoassay to assess vaccine efficacy, which I don't recommend, you should use an assay that specifically targets the trimeric spike glycoprotein conformation, the S1, S2 subunit, or the RBD, since these tend to best reflect the burden of neutralizing potential. You should then, the use of, for example, anti-spike immunoassays, this is a good point, is mandatory in those who receive the mRNA and DNA vaccines, because as I pointed out, the mRNA vaccine only codes for the spike protein. So, for example, you can't use an assay that's targeting the nucleocapsid protein. No clear evidence has been provided on which antibodies assessment, total or IgG, we would prefer both for the purposes of serology testing in recipients of COVID-19 vaccination, such that definitive recommendations on this matter cannot be made. That said, the measurement of anti-SARS-CoV-2 IgM antibodies, though, in this setting has been ruled out by the IFCC due to their questionable protective role, the only modest increase we see in IgM, and their fast decay. And consideration should be given to the measurement of anti-SARS-CoV-2 IgA, since this antibody actually may reflect the efficiency of mucosal protection against SARS-CoV-2 infection. So I think when we think about IgA, it probably gives us a better idea, and this is more of a hypothesis, that it protects better against infection, whereas IgG probably protects better against development of severe disease. And we measured IgA using the Euroimmun anti-spike S1 IgA. We found, again, a modest increase in IgA levels after vaccination. Again, not the same that we'd see with IgG, but that's expected. We did see a modest increase after the first and second dose as compared to baseline. Another important question is the choice between a qualitative, semi-qualitative, or quantitative serological anti-SARS-CoV-2 immunoassay. Preference should be given to automated techniques that provide an accurate quantitative measure of the antibody titer, thus allowing for straightforward and accurate monitoring of anti-SARS-CoV-2 antibodies decay over time. So it's really important to be using now a quantitative assay. Serology testing strategies are challenged by the need for standardization and harmonization initiatives, wherein different immunoassays detect different immunoglobulins targeting different viral epitopes and are characterized by heterogeneous detection limits, different cutoffs to similar ranges of antibody concentration. This is a major hurdle in serologic monitoring programs and any approach for personalized vaccination. Although an international standard was recently introduced by the World Health Organization, which enables test reporting units and test reporting results in binding antibody units per ml, acceptable harmonization of the many commercially available immunoassays is very challenging. And this has been significantly talked about, and the WHO has now advocated for this universal standard. But just to give you an idea, when I checked this morning on the Arizona State University COVID-19 Diagnostic Commons, where they monitor development of all diagnostic assays, to date, there's over 1,000 different antibody assays available out there on the market. So it's quite a large number. So a few technical and logistical considerations for serological testing in vaccinated individuals. Again, like I said, we're going to prefer immunoassays detecting and targeting the trimeric spike glycoprotein conformation, the S1, S2 subunit, or the RBD. We're going to use methods that generate quantitative measures. Again, it's important to always use the same method for longitudinal monitoring, right? Because we've seen some differences in different assays, and you want to have that consistency. And of course, we're going to prefer laboratory assessments, so no rapid antibody tests for serologic monitoring programs for many reasons. I always say it's best to use a multiple assay panel for clinical testing to provide really informative results. It's very difficult to interpret the results. Clinicians are always asking, well, what does this mean? Does my patient have protection? And it's really hard to tell. But kind of using multiple tests will really give us a better idea of that. So we can use an anti-spike IgG, I think it's the bread and butter assay, I should say, and that should give us a quantifiable titer. And then you might want to add in an anti-nuclear capsid IgG. And this won't detect vaccination, but it will give you an idea of whether or not that patient has gone through breakthrough infection, probably important for kids with the multiple inflammatory syndrome associated with COVID-19. You may want to include an anti-spike IgA, maybe in patients who, for example, transplant patients and those who are immunosuppressed. This is a good surrogate for mucosal immunity. And then you may want to do a neutralization test, especially in vulnerable populations, because that really gives you an idea of the actual neutralizing potential of the antibodies that were detected on your first test, which is the anti-spike. So this gives us a real idea. IgM, it's often asked for by clinicians because I think they're used to seeing IgM on their serology test, but overall, it's very hard to interpret IgM as a result because of the fact that it has only mild protection and rather rapid decay. So overall, I think you really need to tailor your testing plan to specific patient populations that you're working with. You may have to have multiple panels, multiple offerings, to best provide for specific patient groups. So what are some of the drawbacks to serology testing? Equivalence between anti-SARS-CoV-2 antibody levels and vaccine efficacy is not straightforward. This is because the role played by cellular immunity and memory B cells cannot be ascertained through serologic testing, as we talked about, right? So you'd have to use different gamma-releasing assays or flow cytometry to assess B cells. Therefore, although the cases of SARS-CoV-2 reinfection due to waning of humoral immunity, variants of concern, or both are increasingly reported, more research is really needed to answer, I think, three key questions. First, what is the individual protective threshold level of antibodies below which the humoral defense against SARS-CoV-2 variants is more likely to fail? We need to know that. We need to know whether memory B cells primed by previous infection or COVID vaccination can be rapidly reactivated and will be capable of generating an efficient anti-SARS-CoV-2 antibody level upon re-exposure. And finally, what is the role of cell-mediated immunity in protecting from SARS-CoV-2 infection, especially in averting the risk of developing severe or critical forms of the disease? Besides the still uncertain relationship of vaccine efficacy, evidence has also been published showing that the different commercially available immunoassays display variable agreements with neutralizing tests, sort of complicating the entire picture, which is again why I recommend using multiple panels of assays with some type of surrogate viral neutralization test. So finally, what are the advantages and drawbacks? I think the big advantages is we can have a program that allows us to track and personalize vaccination, know when to provide boosters, and provides lots of data about the population and able to tailor healthcare according to individuals via serologic monitoring programs. We're able to identify lower non-responders after vaccination and provide them boosters earlier than we would the general population. And again, detect, like I said, the timely detection of fast decay of antibody levels, especially when we're dealing with surges and an unpredictable pattern of this disease. The drawbacks, like we said, there's no equivalence to date between antibody titer and vaccine efficacy or clinical protection, right? We have an idea. There's been some studies, like the one we pointed out from Israel, where they showed the antibody levels related to viral load, but nothing definitive yet. But it is probably our best measure. Again, the measurement of serology is not the same as measuring neutralizing activity, and you need a different assay for that. And again, it's tricky because we don't know how that would then cross over if we did neutralizing activity, say how that definitely goes over to, for example, the variants of concern circulating in your particular region around the world. Cost effectiveness of serologic monitoring studies, though serology is rather cheap, $1 to $5, it still needs to be proven. We need plans to effectively institute these types of programs. And finally, again, probably the biggest burden is that this puts an enormous amount of volume of work on laboratories all around the world. So what are the challenges ahead? I think, like you said, we need to answer those key questions. What titer is associated with protection and protection against variants of concern? And to really answer these key questions, it requires significant collaboration between clinical laboratorians, research scientists, and industry partners. So I advise you to please engage in that process. Feel free to reach out and collaborate because these key questions, we really need answers, right? More variants of concern, I think we'll see them. Variants of high consequence, I'm not sure, but we're certainly going to see more variants, I think, in the coming months. And this will likely lead to more surges, especially going into winter. More people are indoors. And I think the biggest thing is we're going to see an increasing role of labs in monitoring vaccine efficacy in the coming months. I think we're going to see more serological monitoring programs coming. And I think these are going to be really playing an important role in guiding vaccination strategies, booster strategies in the coming months. And I think at the end of the day, I think the point I really want to make is that widespread vaccine stewardship and serological monitoring likely represent our quickest route back to normalcy. And finally, one other point I'd like to add in is that remember that educated ignorance is not the same as ignorance. Saying you don't know is okay with COVID-19. So there's much that we don't know. And saying you don't know because no one knows is different than saying you don't know because you haven't looked into it. So don't be afraid to say I don't know. I think overall, as the country, as a world, we'd be better off if our public health officials just were straight up and said, hey, we don't know. So I thank you all for your attention. I wish you all good fortune in your COVID battles to come. And if you have any further questions, feel free to put them into the chat or send me an email. And with that, I will turn it back over to you guys.
Thank you so much. That was a very insightful presentation. I think everybody in the audience would agree. The questions coming in the chat box. So in case many of you have questions, please type them in the chat box for Dr. Henry. We'll have a panel discussion once the second speaker has completed the presentation. So with that, I think now it's time for me to introduce our second speaker. So our second speaker is Dr. Maite Sabalza. Dr. Sabalza has earned her PhD in plant biotechnology, and her dissertation work focused on the production of human neutralizing antibodies in plants to prevent HIV infection. During her postdoc, she investigated new treatments to inhibit human cytomegalovirus infections. Subsequently, she joined NYU, where she bridged the bench to bedside gap by developing novel assays for emerging viral infections such as Zika and dengue. Currently, Maite is a member of our scientific affairs team at Euroimmun US, mainly focused on expanding the credibility of our infectious disease products. So we welcome Dr. Sabalsa, and I'll pass it over to you, and you can begin your presentation whenever you're ready. Thank you.
Hello, everyone. Aisu, thank you very much for the introduction. And Dr. Henry, thank you very much for such a great presentation. So now please give me some seconds while I'm sharing my screen. Okay. So in this presentation, I'm going to expand on some of the topics that were described by Dr. Henry with key scientific publications. These are my disclosures. And today we are going to start with a general overview of Euroimmun SARS-CoV-2 portfolio as an example of assays that are available commercially to detect humoral and cellular immune responses. Then we will focus on key scientific publications where these assays are used, and we will finalize with a summary with the key points from these publications. At Euroimmun, we offer a complete solution for detection of SARS-CoV-2 infection. We have products for direct detection of the virus as well as products for the immune response detection. For direct detection, we have two molecular assays based on real-time RT-PCR as well as one antigen ELISA. And for the immune response detection, we have different ELISAs to detect different types of antibodies as well as the T-cell response. As we have people in the audience from the US as well as other parts of the world, I would like to clarify that the anti-SARS-CoV-2 S1 curve ELISA is the same as Quantivac, but in the US we changed the name because it was a requirement by the FDA when we received the EUA. So in the next publications, I'm going to discuss how these assays can be used as well as from other manufacturers. The first publication is from Tretrin et al., and the goal was to evaluate the antibody concentration as a marker of the humoral response and the concentration of interferon gamma as an indicator of cellular response after infection or vaccination with the Pfizer-BioNTech vaccine. In these two graphs, we can see the IgG concentration. So on the top graph in blue, we have patients that received the vaccine, but they were not infected before with SARS-CoV-2. And in green, we can see patients that were infected with SARS-CoV-2 and then get vaccinated. We have in the x-axis different groups. We have unvaccinated and different time points after the first dose and after the second dose, and the same for patients with a prior COVID-19. So these results showed that the IgG concentrations reported as binding antibodies units per ml range from 0 to 38,400 binding antibody units per ml. The highest levels were found in the group of patients that had a prior SARS-CoV-2 infection and were vaccinated. In seronegative subjects, those in blue, in the third week after the second dose, the IgG levels were much higher than the IgG levels for those patients that were infected but not vaccinated. Therefore, here we can see that there is a benefit of getting vaccinated when you have been infected. In addition to that, an interesting observation was that all people over the age of 65 had a slightly lower antibody titers, but the difference was more noticeable after the age of 80. And this is relevant to what Dr. Henry discussed in his presentation. And what about the T-cell response? In this graph, we can see the correlation of the antibody response in blue and the T-cell response measuring the interferon gamma in green. We have here the concentration, and here we have three groups. The first one is unvaccinated group that had a prior SARS-CoV-2 infection. Here we have vaccinated with without a prior SARS-CoV-2 infection. And here we have vaccinated plus a prior SARS-CoV-2 infection. So we can see that there is a positive correlation between humoral and cellular immune responses. And the cellular immune response was detected in all the subjects. In terms of concentration, we can see that in patients that were only infected but not vaccinated, the interferon gamma concentration or T cell response was lower than those patients that were vaccinated. The next publication is by Fabricius et al. And the goal of this study was to investigate the cellular and humoral immune response in individuals vaccinated with the mRNA vaccines from Pfizer or Moderna and the vector vaccine from AstraZeneca in the context of previous SARS-CoV-2 infection, as well as homologous or heterologous vaccination. And this is very relevant for the US because yesterday the FDA authorized the a heterologous booster dose, meaning that we can mix different vaccines that are authorized in the US for the booster. In Europe, it has been authorized for many months. Okay. So here we can see the results for the IgG detection, IgA detection, and the neutralization capacity. In each graph, we have 4 curves. In green, we have the response to the Pfizer vaccine. In blue, the response to the Moderna vaccine. In red, the response to the AstraZeneca vaccine. And in gray, we have the response to SARS-CoV infection without vaccine. So we can see that there is the same pattern in the 3 graphs for IgG detection, IgA, and neutralization. There is a difference between the vaccine type, and there are no significant differences between the mRNA vaccines. However, in the case of AstraZeneca, the antibody concentration as well as the neutralization capacity was lower compared to the mRNA vaccines. What about the heterologous vaccinations? So here we have the same IgG detection, IgA detection, and neutralization capacity. In red, we have the results for homologous vaccination of AstraZeneca. In blue, combination of the first dose of AstraZeneca and the Moderna vaccine. And in green, the combination of first dose of AstraZeneca and second dose from Pfizer. So we can see that the antibody titers as well as the neutralization capacity is much higher when using a heterologous vaccination. In this study, it was also investigated the neutralization capacity of the different vaccines as well as different combinations of the vaccine with the variants of concern. So here we have the results, the neutralization capacity against the wild type, the alpha, beta, and gamma variants, and the different types of vaccines used, as well as only infected individuals. So we can see that there is a general trend. The neutralization capacity is lower, significantly lower when compared to the wild type. It happens with all the type of vaccines as well as in infection. What is very interesting is that in the case of a heterologous vaccine, the neutralization capacity was the highest. And what about the interferon gamma or the T cell response? So here we have, again, different vaccine types, different types of doses, as well as the infected individuals. And we can see that there is the same pattern as for antibodies. The T cell response is different depending on the vaccine that the patient received. But in general, the mRNA vaccines had a high level of interferon gamma release compared to the vector vaccine. And also what is interesting is in a heterologous vaccine using AstraZeneca as the first dose and Pfizer as the second dose, the interferon gamma, the highest level was achieved. So in these 2 publications, we were describing the general population, but what about immunocompromised individuals? How is the humoral and the T cell response? Smith et al. In this study assessed the vaccine-induced humoral and cellular immune response in solid organ transplant recipients in comparison with healthy age-matched control group. So here we can see the results for the antibody concentrations concentration for IgG reported as binding antibody units per ml, and the neutralization activity. We have results for after the first dose and after the second dose. And we have three groups: the control group, the solid organ transplant, and solid organ transplant that were infected. So we can see that the IgG levels were significantly higher among immunocompetent controls than in transplant patients after the first dose and after the second dose. And we see the same for neutralizing antibodies. The control group after the second dose had a higher neutralization activity than the transplant patients. What about the T cell response? So here we can see the results after the second dose. So for the controls, the T cell response was detected in 95.7% of the individuals, whereas in the transplant recipients, it was detected only in 64.7. So as we saw in the antibodies, also the T cell response, it was lower. What is interesting is if we compare to the antibody response, antibodies were only detected in 35.3% of solid organ transplant patients, whereas the cellular immunity was more frequently detected in 64.7%. Therefore, the authors of this study concluded that combined analysis of humoral and cellular immunity improves the identification of vaccine responders among immunocompromised individuals. So taking all together, the key points of these three publications are that there is a positive correlation between humoral and cellular immune responses after SARS-CoV-2 infection and or vaccination. The humoral and cellular immune responses are higher after first dose of vaccine in individuals that were undergone SARS-CoV-2 infection. And combined analysis of humoral and cellular immune responses improves the identification of vaccine responders among immunocompromised individuals. Also, something very important is that the strength, the breadth of the immune responses, as well as the neutralization activity, are influenced by the type of vaccine and the use of heterologous or homologous vaccination regimes, as well as the presence or absence of a previous SARS-CoV-2 infection. And finally, we can see that there are available several commercial assays that can be used to monitor the humoral and cellular immune responses. And with that, I'm going to thank you for your attention, and I'm going to pass control to my colleague Aya. Okay.
Okay, thank you, Dr. Sabalza and Dr. Henry, for two wonderful presentations. As we progress further in the pandemic, we're getting a lot of necessary answers based on ongoing research, but we're also discovering a lot more key questions. So with that said, we're going to go into our Q&A panel discussion. I see Dr. Henry has joined us. Perfect. So our first question came from a patient. So this patient is B-cell compromised. They're on rituximab infusions every 4 months. She states that after 2 Pfizer vaccines, she did a spike protein antibody test that showed 0 antibodies. She did a 3rd dose with Moderna now. And, she's wondering first, what are your thoughts on the likelihood of having any immunity? And do you think that, what kind of antibody test would you recommend for her? She is living in Canada currently. Dr. Henry, this was for you.
Sure. I think, these are the types of patients that, I think everyone has, the greatest concern over. And it's also because of patients like that we, we still need to have protection, have social distancing measures, keep wearing masks, because we haven't reached that herd immunity threshold. So these patients are very important. And it's important that we protect our vulnerable patients. I think in this case, trying to get a neutralizing activity assay would probably be the best bet. again, this is why I talk about that we, really need to engage in sort of that multi-panel in terms of healthcare professionals. We need to use multi-panels, especially for vulnerable patients. But in this case, this is the type of situation where, using that like surrogate viral neutralization test would be, beneficial, because that would really give us the idea that we might not, maybe you don't have a high titer, but maybe you do have antibodies that are, very effective at neutralizing the virus upon exposure. Of course, those are still hard to come by out there. There's only a few on the market and limited places that have those. We're working hard to get it into our lab. But until those are available, I think in her cases, what I would recommend would be really repetitive, serology testing with an anti-spike type of IgG assay. That type of patient may have to have it every few weeks. Those who are very immunosuppressed might have to have it every couple of weeks, every other week, until we, are able to, build up that immunity or offer better, forms of testing out there.
Thank you. So based on your knowledge, is there any test available that measures spike-specific memory B cells, and would you recommend testing for it at this time?
I believe that, I think that the only way to measure that currently is with flow. I don't know if there's an alternative option. I don't think there is. So that would only have to be done at, very specialized labs as of present. But it's definitely something, that hospitals that are dealing with immunocompromised patients should really consider moving forward. Again, offering that multiple panel of tests, that includes both T cell assays, memory B cell assays in very specific populations. I think it's very clear this isn't going away. And I think clinical labs need to start to offer a wider comprehensive panel of testing for patients who are, immunocompromised or at very high risk, children as well, in order, to, provide that type of information, and give patients reassurance and, clinicians reassurance, hospitals reassurance on the protocols they're using to protect the population.
Okay. The next question, I know you mentioned that antibody response is high in younger populations. So does it raise any concerns for giving the vaccine to children between the ages of 5 and 12, especially, that the younger range of that group?
I actually think that the opposite. I think that's great news that the antibody levels are so high. Probably means then, that, because of that, they will need less frequent boosters than say adults. So I think that's very good news. I think the clinical trials that have come out, the data that's come out have shown that, I think Pfizer is 93% effective of preventing serious infection in children, which is just fantastic news. I think we need to vaccinate kids as soon as possible. So I hope the FDA moves on it. I think, there's still concerns, for example, with children about, the long-term effects of COVID. So having that high titer, especially the very high titers we see, it's probably great for getting kids back in schools. It probably will correlate with, the fact that they'll have not just decrease of severe disease, but the higher the titer, I think it's safe to assume, we don't have data to say, but assume that also means a lower threshold for infection. It means you're not spreading the virus back and forth when they're at school all day. So I think that's really great news. And I don't think there's any safety concern, for example, with the high titer.
Perfect, thank you. And so going into booster shots a little bit, couple questions here. The first one is about multiple sclerosis patients. Should they be prioritized for these boosters? What are your thoughts on that?
Yeah, and I think actually we did have a study on that included patients with MS, and I probably skipped over it because we had so many comorbidities that have been out there. But absolutely, patients with MS, they're often on immunomodulating medications. These patients should definitely be in the group of patients that are prioritized for boosters.
Okay, and regarding like a low antibody test that was received recently, do you think that is any indication of getting the booster? Or I know we don't know much about what the values mean at this time, but if a patient gets a low antibody test, would you recommend a booster at this time?
Yeah, absolutely. I think anyone who's been gotten, within over 4 to 5 months, I think CDC's guidelines are 6 months, should definitely, try to get that booster as soon as they're eligible to do so. Again, we don't know what that cutoff is, and that's why I do recommend, having a monitoring program, doing a 1 month, a 3 month, a 6 month assessment, right? Especially vulnerable populations, so we can kind of see, your antibodies level dropped to tenfold. We don't know what that protection is, but to me, I would say I don't like that. I probably want to go and get a booster. If you have an antibody test and it's coming back low, on the ranges reported by most immunoassays. I think that's also an indication, like, you don't want to be on the low end, right? I think that's an indication. It's not absolute. I can't say anything for certain, but if I came back and it was on the lower end, towards the cutoff for positivity, I would definitely say, hey, you probably should get, I would get the booster if I was that patient.
Awesome. Next question is, I know you mentioned using a multi-assay panel for clinical testing. The question is, do you know if anyone is currently running an IgA test, and when would you recommend that test?
We know in-house we have developed, a particular IgA test. I think the research there is, still forthcoming, but I think there is data that shows that, IgA really correlates well with mucosal immunity. When we think about the immunoglobulins, right? IgA's real job is to protect us at the mucosal level. There is circulating IgA, it's a small quantity, but most of the IgA, sits, along our respiratory tract, in your nasal mucosa, in your GI tract, right? And its job is to bind up foreign invaders, upon first exposure. So I think, like I said, that IgA, I think we should see it, start to be more incorporated into, regular routine testing, because I think it does give, look, there's no perfect answer. There's no, like, you're going to look at this and know exactly what your patient, what type of protection they have. But looking at multiple things, I think, can start to give you a better idea. And looking at your IgA might tell you, hey, this patient, they have pretty good protection against, the chance of infection. And then you can look at IgG and be like, well, they have pretty good protection against, severe disease. And I think starting to use these different combinations of things gives us much more insight. So I hope IgA gets much more incorporated into clinical testing programs henceforth. And we've actually showed, and others now, that, there is an IgA response, after the vaccination, which is great news. We'll kind of have to see how that plays out with breakthrough infections as well. And, the more we get in these laboratories, the more we can get, research programs going as well, to answer these important questions.
Yeah, I would like also to comment on this. Henry, I totally agree with you. Our IgA ELISA was one of the first ones that we developed with the IgG, because we know that in respiratory infections, IgA is a good marker for the early phase of the infection. In the US, for us, it's for research use only, but I know that in other parts of the world, the IgA ELISA is used and is very helpful, because, as you mentioned, we need the more information, the better to understand what's going on. Yeah.
Thank you. Okay, next question. Dr. Henry, I know you mentioned that the CDC, in your opinion, made a mistake on not measuring breakthrough infections. So what would be your recommendations to analyze vaccine efficacy for CDC or the vaccine manufacturers?
Well, I think, outside of the vaccine manufacturers, I think it's good to have, labs around the world, that are doing independent monitoring of vaccine efficacy, right? We really sit at that sort of interface, right, between the patient and science. And, we should take advantage of that, to further our understanding of this. I think that we do need widespread serological monitoring programs. I know in the US, I think one state has started it, which is West Virginia, is going to start doing, testing across their population in their state. But we need those types of programs to really understand, what type of immunity that we have across the population. That can really, help guide our public health knowledge, understand breakthrough infections. But it's tricky, we need to make sure that, public health programs are then utilizing the same assay, right? Especially because they don't have standardization between assays. So you need high throughput technologies, to be employed, and you have to use quantifiable. You need to use quantifiable results that are quantifiable in order to, be able to track that over time. So you can't use any semi-quantitative or non-quantitative methods. So we really need a quantitative method. So I think you need to start, I think public health agencies should prioritize these programs. I think they need to prioritize, regular testing intervals. And like I said, I think this is really the best option we have to our route to normalcy. It's going to be the combination of boosters and serological monitoring programs. I don't think one without the other is going to be efficacious. And I think that's the problem we have now. So I think, especially now that boosters are approved, I think we need to start, combining these methods to really get us, moving forward and get us to the end of the pandemic.
Thank you, Dr. Henry. There's a follow-up opinion question as well. So currently the CDC does not recommend anybody testing post-vaccination. What are your thoughts on that?
I strongly disagree with the CDC. I think my opinion on this is well known. I don't know why. I've read their explanations. I don't think it makes much sense in my opinion. I think we should have serological monitoring. I think the fact that we need boosters goes to show the fact that we should have serological monitoring. I don't think you can say that you need to have boosters, but then we shouldn't, have serological monitoring and recommend testing, especially in vulnerable populations. And I know from my experience, most clinicians, and especially those who deal with those vulnerable populations, are routinely ordering those tests in their patients for that very purpose. So I'm not sure where why they're sticking by that particular guideline, but I think the evidence is quite clear. There's significant individual variability in vaccine responses, and we should be trying to measure those and assessing those and giving boosters that are, oriented towards the patient, guiding, give that personalization of care. And we can't do that without, monitoring programs. And I think some state agencies, like I said, are taking their own initiative to do that. And I think, I would hope that the CDC really strongly considers changing that particular, recommendation, because I think the data is quite clear. When we see those inter-individual responses, we see the differences and so many factors influencing it, that monitoring, especially in highly vulnerable populations, is extremely beneficial.
Yes, definitely. Thank you so much for that. This is a follow-up question to our patient who is on rituximab infusions.
Mm-hmm.
So she's wondering if repetitive testing with the anti-spike IgG assay shows no antibodies, what would you recommend at that point? Repeated vaccinations or something else?
I think it's hard, especially in a patient who's on a drug like rituximab. I'm not an expert. I'm not an oncologist. So I think, she needs to talk to her clinicians. I think the issue is that a lot of people have, and I get lots of questions, that they go to their clinicians and unfortunately they don't have an answer. They don't know themselves. And it's not that they haven't looked into it. There just isn't that data. And I don't think, for that patient, there is that, particular good answer. I think the best thing I could tell that patient is, continue to take your preventative measures, continue to socially distance, keep a bubble, ask people who are going to come around you to take a test, at least a rapid test before coming to visit, or you go to a family party. Everyone's safer when we work together. And I think we have to really wait to see what kind of data, that is. If you've already received the booster and you're still struggling to see responses, that's more tricky, maybe again, talking about getting a broader range of testing from a more advanced clinical laboratory that can assess T cells and other things like that might be a good option in that particular patient. But again, I don't like to say that we don't know, but we don't know the best option for a patient like that. We're working at it, but for now, keep your protective measures, continue to talk to your doctors, and hopefully we'll have, better options moving forward.
Yeah, I can add something to this. We have ongoing collaborations with institutions performing some research on cancer patients and see how the T cell response, our assay can work. And there are other studies going on. So I hope we will have more answers in the near future. Thank you.
Perfect. Thank you. This next question is for Dr. Henry. So Dr. Henry, are you advocating using immune monitoring as a method of determining when a booster is necessary? And other than, having that crucial information being good to know for future research, what clinical reason would doctors have to order that test and insurance pay for it?
Well, I think, yeah, I'm absolutely, advocating for that. I think we still, I think it has to be something where we're doing it and using that data at the same time to refine these types of programs, right? It isn't going to be a situation where we can do studies monitoring, serology for a year and then decide later how to build a program off of that. These things need to go hand in hand as we move forward. So we need to have serological monitoring that acts both for generating research data to guide the program further, while also providing important data about when we can initiate boosters. Again, we don't have those thresholds, so I think that is what we need to establish within those programs. But I think that the justification for that, I think that's something we should look into. By the way, it should be publicly health funded, right? Serological monitoring is cheap. It's much cheaper to do serology assays on our population than it is to shut down our economy. Yeah, I think that's, if you're actually about public health costs, it's cheap to do serology assays. It's cheap, so I think that, for that cost, in terms of justification, I think the justification is very clear, for those who are over the age of 65. I think they're, those who have comorbidities. I think the justification is very clear about why we would need, as from a clinician side, for insurances to cover that type of testing. I think the evidence is there for that, despite the fact that the recommendations are not. So, in terms of that, absolutely, I think that, there is that justification for insurance companies to cover that and to use it to guide vaccine booster administration. Again, but we're talking about an era, and like I said at the beginning, anything I say can be wrong. We're talking about an era where we don't have the type of. It's not like I'm sitting here going through these long years worth of clinical data, to give this type of information. We have to work with the best data that we have right now, right? And I think that's the approach we need to take. And I think some of the fact that we haven't used that approach has been detrimental to our public health response. We need to work with what we have, and the best option we have is using these types of serological monitoring, to really guide us forward. And I think that's what's been missing overall in our current, vaccination programs.
Perfect, thank you. So we'll end on one more question here. So currently there are semi-quant IgG and total antibody assays, but no semi-quant IgA assays, since they're all qualitative at this time. What are your thoughts on how qualitative assays are more or less helpful than a semi-quantitative assay?
You want me or you take this one?
Either one.
Do you want to answer? Or you
Uh,
want me?
yes, I thought you were going to answer, Dr.
Okay,
Henry.
no,
So,
okay.
okay, yeah. So with quantitative assays, the estimation of the antibody concentration is more accurate than qualitative. So it depends also on what's the goal. If we want just to see if there is an antibody response, then with qualitative will be enough. But if we want to measure the antibody concentration after vaccination, in this case, it's better to have a semi-quantitative or quantitative assay.
Yeah, I agree with that.
All right, and one quick question that just came up was, what is the protective level, if any, of neutralizing antibodies in vaccinated individuals?
I think that's like one of the questions we have to answer is what is that, threshold for protection? Especially when it gets more complicated when we're talking about the variants, right? Because we start to see that decline in that neutralizing activity. So I think these are all questions we need to answer. But the only way we're going to answer those questions is with lots of data. That's what we need, like I said, serological monitoring for both to guide and to, be able to answer those questions, right? Because clinicians are, I get asked that all the time is, what does this mean? And I don't have an answer because, I could say, you may have protection, you may not. It may protect you against certain variants, it may not. We don't know. And that's why we need these types of programs in place so we can, start to answer, these questions. There's so many variables. There's so many patient level factors, as you've seen, that we talked about in both of our talks that come into play with impacting this. So, we need data, and that's why we need these, these types of programs. Well, I'm such a strong advocate for them. I have lots of respect for everyone at the CDC, but, I wish, we would have done things differently, and I wish moving forward we would start to do things differently. So,
yeah, and I also want to add that it's very important to have a good international standard in order to be able to harmonize the results, and like that we can take conclusions, we can compare results from different manufacturers and see to better understand the immune protection.
All right, thank you, Dr. Henry, Dr. Sabalza. So this concludes the first episode of our infectious disease webinar series. After this webinar, a short survey will pop up, and you can provide feedback and also take a short quiz to redeem your PACE certificate if you're interested. Please stay tuned for our next webinar, which will be in early December on tick-borne diseases, the emergence of Powassan virus, and current diagnostic testing options. So thank you again, and we look forward to seeing you at the next webinar. Take care.